Welcome everyone to the today's lecture. So today we would be discussing about the concept of vaccines, what are their different types, and the process of vaccination. The main learning objective of this lecture is you will be getting to, understand the concept of vaccines, that why they are needed for the humans, why everyone is after the vaccine that they get, vaccinated, and what is the need, what are the different type of vaccines that are available, how it was developed, what were the milestones in the discovery of these vaccines. At the end of this lecture, you'll be, able to learn the importance of vaccines in our life, why it is so required so as to, gain acquired immunity, what are the mechanism of action of different type of vaccines that we are currently using. For example, recombinant vaccines, live, attenuated, toxoid, or edible vaccines also, and what are their developmental procedures and what sort of companies are basically playing a role in it in the development of these vaccines. So starting with a brief introduction part, first of all you need to understand the difference between epidemic versus pandemic. Epidemic, it is an event whenever a disease is actively spreading and that disease is sometimes out of control in a particular area, that is known as epidemic. However, if you talk about pandemic, it relates to the spread of a particular disease that is out of control, but that is at geographical level. You can say it is distributed or it is being spreading to wide range of geographical areas. That means that can affect the whole country or whole world. That is known as pandemic. Now, there are many pandemics and notable epidemics throughout the history. However, all of us are quite aware of these pandemic and epidemic situations in the current COVID scenario. But I would like to throw a brief light on the history of it. The first of all, the plague comes whenever the Europe's population had dropped around 50%. That means 50% of Europe population was, depleted because of this plague. Then comes the typhus that happens during the World War One, in which around 1.5 lakh people were, destined to death in Serbia. Then smallpox, malaria, tuberculosis, and their, the death rate of these diseases, the death rate was increasing day by day. However, there was no efficient solutions to it. For example, in the cholera epidemic, in that case, around 20,000 people were died because of the cholera in the year 1907 to 1908 in the Makkah. And this is a real picture of that. Then is influenza epidemic that happened at that time. Around one third of the world population was infected due to influenza virus during the 1918. At that time also there was a very critical situation because there was no possible or effective solution available with that. And talking about COVID-19 pandemic, the worst pandemic in the history, and I think everybody is quite aware of it. That there was also many news channels were circulating the same information also, that this pandemic situation has happened after 100 years, because in the year around 1920 the influenza pandemic was happened. So this is the worst pandemic in history because the total cases and the death due to the COVID-19, due to this coronavirus, this was quite, it was claiming many lives. You can say it has claimed so much of lives that the even the WHO is quite worried about it, that how they can cater this infection. That is why the process of vaccination or vaccination drive all around the world is going on. It's doing rounds on greater basis. So these are the death tolls because of different epidemic and pandemics throughout the history. As you can see, Black Death around 200 billion, then smallpox around 56 millions. In the 17th century, plague was happened. At that time, 3 million lives were claimed. So going on with that, SARS was there in the year 2003. It claimed around 770. So there are the death toll because of these epidemics and pandemics is quite increasing, and there we must have a efficient solution, an effective solution to cater these epidemic and pandemic situation. In that case, in the current COVID scenario, right now, our current new normal of life is our handshakes are, are changed. We are using alternatives. Either we are waving our hand, we are, elbow bump is being going on, or namaste is quite in trend. And, all of these things, however, if you talk about it, it's around almost been 1 year that COVID-19 pandemic is still here all around the world, and everybody is worried about it. Every agency, WHO, or every country's the main government is worried about it, how to cater it. That is why the vaccination drive is on full swing. So, are we going to stay like this, or we shall opt for a possible solution so as to cater these pandemic and epidemic situations? In that case, the main solution to it is get yourself vaccinated. You can say vaccination. Million of people die every year due to range of microbial infections. In that case, the most effective way to save the humanity from this is to undergo vaccination. So, what is a vaccine then? If you're talking about a vaccination, then what is a vaccine? Vaccine basically it is a biological preparation that provide the host body immunity to a particular disease. It basically contains a agent, or you can say infectious entity, microbe, their whole cell part, or their small material of it that resembles a disease causing organism, and it is often made from the weakened or kill form of the microbes. So that means it is basically the part of microorganism in which its pathogenicity or virulence had either it has been weakened or it has been killed. It can be the toxins produced by that very microbe, or it can be the surface protein, or it can be the whole microorganisms also. So that such kind of agents, what they basically does, they stimulate the body's immune system to recognize that antigen as foreign. That means as non-self. Then after recognizing that material non-self, our innate immunity, adaptive immunity, all of our immune cells, they come into charge and they will, destroy that very agent, and will also store a memory. That means they will keep a record of it, that in case the future encounter is happening, then we shall be prepared for it. So, in that case, the immune system, in case of secondary encounter, they can more easily recognize and destroy any of these microorganisms that it later encounters. Now, this term vaccine and vaccination, they are basically derived from variola vaccine, that is smallpox of the cow. The term was derived, basically devised by Edward Jenner, who did some experiments on the cowpox virus. That if the milkmaids are infected with the cowpox, in that case they are immune from the smallpox, and he even proved that concept by by taking the fresh material from the lesions of cowpox infected person, and they injected that lesion part into a healthy 8-year old boy. And after he developed the symptoms of cowpox, he was well after a few days, but that infection basically provided that boy immunity to smallpox. So after that, this concept, this vaccination and vaccine concept was basically derived. The main mechanism of action of vaccine, whenever you get yourself administered with some vaccine, then how our immune system responds. For example, as you can see, this is the foreign microbe with antigen. In that case, this is the macrophage that will ingest the antigen. That means phagocytosis would be triggered. It will be ingesting it, it will be processing them and present them to the T cells. T cell will differentiate into killer T cell, helper T cell, suppressor, and memory. Helper T cell then will secrete cytokines so as to activate B cells to produce the antibody, and will also activate cytotoxic T cells so as to induce the cytolytic activity. Memory response so that the secondary encounter can be prevented. So that is the concept. Based the main discovery that was done by Edward Jenner, that proved a breakthrough discovery, because of which the era of vaccination and the development of different type of vaccines against different infectious agents and pathogenic microorganisms was mapped. Now that means these vaccines till now they are considered to be the most safest and effective medical intervention available. They have contributed a lot to the steady decline in the mortality and morbidity rate of many diseases that are basically happening due to many kind of infectious organism or pathogenic organisms. The currently available vaccines they can prevent up to 3 million deaths, and many childs are being protected from serious disabilities every year, because they when a kid is born, they normally provide you a complete list and timeline of vaccines that a kid has to get vaccinated with. So these vaccine improvement strategies are also being implemented at a very faster pace, so that the new adjuvants and new kind of strategies can be done so as to improve the efficacy, and a vaccine should have a broad antigenic variety also. So in the past, when this concept was devised, at that time what sort of traditional vaccines we were using, we were in that case the infectious thing that was grown in the animals, for example vaccinia virus, it was in calves for smallpox, rabbit brains for rabies. Simply bacterial cultures were being used, and then they were inactivated, and then they were administering that thing to the humans. So all of these things were traditional practices. In that case, there were certain limitations, that there are certain organisms which you can't grow in the culture medium. And another thing is, the person who is handling all of these culture, the safety is very important. That it was a quite an expensive affair. In that case, if some insufficient attenuation, or the living agent is not completely altered, or the virulence is not completely weakened, in that case it can have some adverse effect. Sometimes some reversion to the infectious state was happening. In that case, that was also a limitation. For the traditional vaccines, you need refrigerations, and in case of all infectious antigens, all infectious agents, it was not sometimes working. However, the yield of vaccines that was coming from the traditional procedures, that was quite low as compared to the modern system. However, large number of antigens, they also stimulate immune system, which is not desirable, because we are using whole cells. We are basically growing in animals, then you are injecting whole cell material. In that case, there can be many antigenic determinants that can activate the immune system in a adverse manner also. So that was the major limitations. If you talk about this, that how the vaccines are made, from what is the material, there can be either live microorganisms, that so that their virulence is reduced or weakened. They can be dead microorganism also, however, in which some part, or some surface protein, some receptor, that can also be used as a vaccine. Now, depending upon this, there were three categories of vaccines: first, second, third. First was when this vaccine was being developed. At that time, whole cells or virus particles were being used so as to treat them as a vaccine. For example, BCG or polio drops. And in case of second generation vaccine, there were scientists were using some specific antigen components of the organisms. You can say surface proteins. In that case, the example is tetanus toxoid, hepatitis B virus vaccine, pneumococcal vaccine, in which the capsular polysaccharide was being used as a vaccine material, as an agent. Then was third generation vaccine, in which the plasmids or genes, that means now we have shifted our concept directly to the genetic things. Now the plasmids or the genes responsible for the antigen formation, that was being used. So example is DNA vaccines. So however, depending upon this, different type of vaccines are there: live attenuated vaccines, recombinant peptide vaccines, recombinant live vaccines, inactivated, subunit, conjugate, edible, dendritic cell, naked DNA, toxoid. We'll be discussing one by one. First is live attenuated vaccine. So as the name suggests, live, that means the cell would be live. attenuated, that means it would be having a live whole cell or virus particle, but their pathogenic ability or their virulence would be weakened in the lab, so that it cannot cause any kind of infection or any kind of disease, because a live attenuated vaccine is the closest to the natural infection. These vaccines are very, good teachers of our immune system. So the immune response that is mounted against these kind of vaccines, that is quite effective. For example, vaccines against measles, mumps, chickenpox, they are all live attenuated vaccines. Second class is inactivated vaccine. As the name suggests, so these are inactivated. That means the researchers or scientists who are producing such kind of vaccines, what they do, they kill the disease causing microbe, either by using some chemicals or by giving some heat treatment or by using some radiation treatment. However, such kind of vaccines, they are quite stable and they are quite safe, because there is no, danger of reverting back to the infectious state, because the organism is completely killed, because they can't mutate back to the disease causing state or infectious state. Example is vaccine against influenza, polio, hepatitis A, and rabies. So, talking about the other types, recombinant vaccines, there are many types: subunit, DNA, conjugate, edible. However, the recombinant vaccines are considered to be safer and more effective vaccines, in which first is subunit vaccines. In the subunit vaccine, instead of the entire microbe, this, in case of this, only the antigenic determinant that can best stimulate the immune system of the body, that was being used as an agent to prepare the vaccine. However, in some cases, these vaccines use such kind of epitopes that are quite specific to the certain parts of antigens that antibodies or T cells recognize. So they contain the essential antigens or antigenic determinants or specific antigenic determinants also, so they are also quite effective vaccines that are being developed. For example, for the plague immunization, subunit vaccines are being used. However, this complete thing that how we can develop, how we can use the subunit vaccine, it was, basically confirmed by certain experiments in which it was shown that the capsid or envelope proteins are enough to, initiate or mount the immune response. For example, in case of herpes simplex virus, envelope glycoprotein O, it can, initiate quite strong immune response. In foot and mouth disease, the capsid protein VP1 is responsible. Extracellular proteins that are released by Mycobacterium tuberculosis, that is quite effective in, mounting the immune response. So these antigenic determinants, they can be basically used as vaccine material. So there are further types: multivalent subunit vaccines. That means that you can prepare these things by two ways: solid matrix antibody antigen complex or detergent to protein agent. This is the first one, that is SMA, solid matrix antibody antigen. In this is the T cell epitope and B cell epitope. This is a solid matrix onto which monoclonal antibody to a particular antigen is already attached. So this solid matrix antibody, it basically makes a complex whenever the antigenic determinant of that T cell recognizing epitope or B cell, it attaches and it forms a solid matrix antigen. Next is detergent to protein antigens. What happens is detergent extract the membrane antigens or antigenic peptides. So this is the ISCOM preparation, that how we can extract the membrane antigen or antigenic peptides, and how we can deliver that also. So this is the antigenic moiety. This is a liposome formation. This is the detergent plus QLA. The ISCOM formation has happened over here. After, you can see that after the treatment with detergent, micelle formation is happening. This complete encapsulated or, covered antigenic with detergent, it is basically how it is entering inside the cell, that it is entering inside the cell with the help of these ISCOM, and it is injecting the antigen. After that, antigenic peptide is being digested with proteasome complex. It enters into endoplasmic reticulum with the help of TAP transporter. MHC complex recognize those peptides and present onto the surface so that T helper cell can recognize it. Now, Selection and delivery of vaccine peptides. We can use discrete portion of a surface protein. That means certain portions. We can use that. These domains are known as epitopes or antigenic determinants, which will be recognized by the specific antibodies. However, we can use some certain carrier proteins also. Basically, these are small peptides which can't be digested. However, they do not, the immune response against those carrier proteins are also not mounted, because the main concept of using this carrier protein is to provide the stability to the subunit vaccines. Next is recombinant vaccines. That in the case of recombinant vaccine, this is the how this vaccine was basically developed. As you can see, this is the vaccinia virus, and which a portion of genome of this vaccinia virus was taken, and in which hepatitis B virus gene was injected. After that, HSV gene. After that, influenza virus gene, and this vaccinia virus genome was basically containing genome 3 inserted gene. And when this genome was, then the recombinant plasmid was made. When the plasmid was inserted, the recombinant plasmid carrying these 3 genes, it was basically allowed to multiply in E. coli cells so that the selection can be done. In that case, after insertion, when it was multiplied inside the animal cell, the vaccinia virus was, recombinant vaccinia virus was extracted. This is the procedure. Suppose this is the pathogenic microorganism, and onto its surface certain epitopes would be present. That means antigenic determinants. These would be converted into cDNA. Then after that, the cDNA would be inserted inside the expression vector. The gene of this antigenic determinant present in this expression vector would be transfected inside the cell, inside the bacterial cell. After that, the recombinant selection would be done, and once the recombinant cell, that means the bacterial cell expressing the antigenic determinant is selected, then in the large amount that recombinant peptide would be produced in the lab. After that, the purification of recombinant peptide could be done by using many downstreaming processes. For example, isolation of total protein content, then purification with chromatographic techniques, structure prediction. After that, you have got the purified peptide. The structure elucidation is done. In that case, you have to check for the immune reaction, for immunogenicity. In that case, first of all, the vaccine or the purified peptide is administered inside animal models. You can use rabbit. In that case, after everything is okay, then it is ready to administer inside the humans for the vaccination process. So this is the complete chart that how it is developed. However, India is the fourth country after USA, France, and Belgium in the world to develop an indigenous hepatitis B vaccine. However, India is the largest exporter of hepatitis B vaccine. So this is the process of production of hepatitis B virus vaccine. This is the hepatitis B virus genome, DNA, and onto its surface, this is HBs antigen. What happens is, if you want to produce a hepatitis B virus vaccine, in that case, you have to digest this thing with, you have to digest the DNA with the same kind of restriction enzyme, for example, EcoR1. Here we are using yeast vector PMA56, and which after digesting it, this is the gene that will code for this antigen. We will be inserting this gene into this plasmid. This is inserted, this is transformed. As you can see, the recombinant plasmid is ready. This recombinant plasmid carrying HBs antigen gene would be transformed into yeast cell. And then these yeast cells would be grown on the tryptophan-free medium. Why? Because this vector is basically, this transformed cells would only be selected onto the tryptophan-free media. Then you can select the recombinant cells, and these cells then can be lysed so that to isolate the purification of HBs antigen that can be used for the immunization. This is the complete description of it. The gene encoding has been identified. Recombinant hepatitis B vaccine is a subunit vaccine. We can use Saccharomyces cerevisiae as a, for this purpose. The gene is inserted inside the, yeast vector in which ADH gene is present, that is alcohol dehydrogenase gene is present, and to the upstream of the promoter of this gene, the HBS antigen gene is inserted. The cell will be grown in the tryptophan-free medium. It would recombinant cells would be selected and it would be cloned and they would be purified from it. Next is DNA vaccines, however, still in the experimental stages. These vaccines are quite promising and quite effective. They have been tested in humans. They have taken the immunization of to a new technological level, because these vaccines, they are basically dependent upon both the whole cell particles and its part. So that means the main vaccine that has been developed by using this is influenza vaccine. In this case, what is being done? Suppose this is the viral gene. You have what we do. We inject this viral gene inside the expression plasmid. This is a plasmid with a foreign gene, and then that plasmid DNA carrying the foreign gene, the amplification of that plasmid is being carried out. This is the amplified amplification of this recombinant plasmid. After that, this plasmid DNA carrying the viral gene, it would be purified from the bacterial cell in the culture medium, and you can prepare the DNA vaccine from it. This is the complete part. This is the DNA for antigenic creation. That is the plasmid vaccine that is present inside this cell. It will start synthesizing messenger RNA, antigenic protein would be produced, it would be fragmented. After that, it would be presented onto the surface of MHC. Here, the MHC, it would be recognized by T lymphocyte and the response would be triggered. Or we can either, what would be happening, these proteins would be recognized by B lymphocytes and B lymphocytes will start producing antibody against it. However, there are many advantages of using DNA vaccine in immunization, because the tedious and costly processes of purifying antigens or creating recombinant vaccine is not necessary. Because in this case, what we are doing, we are directly, preparing the recombinant plasmid and we are, injecting it. However, they are quite specific in nature because they produce the target proteins. They trigger the specific immune response against a specific pathogen, and the response rate is quite high as compared to other kind of vaccines. And DNA vaccines are found to be quite stable, whether at low temperature or high temperature, rather than conventional vaccines. So these are the methods of delivery. They can be delivered intramuscular, by intramuscular injections, by gene gun delivery method, or by nasal spray. Recombinant vector vaccines, these are the experimental vaccines similar to the DNA vaccines. However, we can use both attenuated or bacterium to introduce microbial DNA to the cells of the body. Here, the vector refers to the virus or bacteria that would be used as a carrier. For example, the DPT vaccine. Next is toxoid vaccines. As the name suggests, toxoid, that means there must be some toxin. That means for bacteria, those bacteria or those infectious organisms that secrete toxins or harmful chemicals, a toxoid vaccine might be the answer, because these vaccines are used when a bacterial toxin released by a, or you can say, or infectious toxin that is being released by a particular kind of microorganism is a major cause of disease. For example, Clostridium. In that case also, scientists have found that what happens is if you inactivate those toxins by treating with some chemicals, for example, formalin, that detoxified toxin can be used as vaccine. Those are known as toxoids, and they are much safer, and they can be used in vaccines. For example, Crotalus atrax toxoid is used to vaccinate dogs against rattlesnake bites. So these are the vaccines which consist of exotoxins that have been inactivated either by heat treatment or by using some chemicals. Basically, the main concept is to build the immunity against those toxins. However, whether the bacteria will be producing it or not. For example, Clostridium botulinum, the botulinum antitoxin, diphtheria antitoxins, all of these things are basically, to provide the immunity against these kind of infections. The toxoid vaccines are developed. Next is conjugate vaccines. For example, if a bacteria is having some outer coating of sugar molecule, you can say polysaccharides, then many harmful bacteria, they possess this polysaccharide layer. In that case, the main, reason why scientists were working in this area. That means these polysaccharides can be used as an, for as an antigenic agent or some agent to stimulate our immune system, because these polysaccharide coatings, they basically disguise a bacterium's antigen so that the immature immune system of infants and younger children can't recognize or respond to them. For example, Haemophilus influenza type B vaccine, that is HIV. However, there are many more vaccines in the pipeline. HIV vaccines, there are many obstacles to it. For example, genomic diversity of HIV strains, the progression of infection despite vigorous immune response, and the good animal model is not there so as to check the immunogenicity, and the potential enhancement of the HIV replication by neutralizing antibodies also. However, there have been many vaccine strategies. For example, whole inactivated cell is being used, live attenuated HIV peptide, live virus, DNA, recombinant protein peptide. However, there is another class of vaccine also, that is tumor vaccines. Vaccination strategies have also, being carried out in case of, for treating cancer, for treating tumors. It is currently in developmental stages, in which such kind of vaccines are being prepared, vaccines containing tumor antigens, such as those associated with, say, prostate cancer. In that case, some prostate specific antigens can be used as a vaccine material, as well as those associated with breast cancer, colon, ovarian cancer. So they are basically offering a new hope for the future. Then there is a edible vaccines. That is a complete new approach to the oral immunization. As the name suggests, edible, that means we can eat it. So in this case, these are antigenic proteins in which we have genetically engineered them into a consumable crop so that everybody can, have it. Following consumption, the protein would be recognized by our immune system as non-self, and strong immune response will be mounted against that. In that case, the first edible vaccine was tried in tobacco. In that case, the patent was also granted in the year 1990. Then this edible vaccine was first of all tested in the humans in the year 1997, when the scientists, they basically asked for many people that they, consume these anti-diarrheal transgenic potatoes. Then first of all it was tried in tobacco, then it was tried in potato. However, there are many candidates for edible vaccines. Potato was the first major system that was being used for vaccine production, so for the human consumption. We can use potato, banana, tomato, lettuce, carrot, rice, wheat, corn, peanuts, soya bean. So all of these are potential, you know, candidates for discovering for the development of edible vaccines. However, there are few edible, ideal properties if you want to develop an edible vaccine. First of all, it should be non-toxic and non-pathogenic in nature. It shall provide a long-lasting immunity, humoral as well as cellular. It should be environmental friendly. That means it shall not cause any kind of harm to the environment. The vaccination procedure should be quite simple. However, it would be quite simple because it is edible. It should be highly effective and cheap. It should be safer for immunocompromised people also, and there shall be no side effects. This is a method of development actually. If you want to develop some, edible vaccine, for example, suppose this is the gene identified and isolated of a particular, infectious thing. In that case, the gene would be replicated. There can be two ways. Gene would be replicated. You coat that gene, coat that DNA with the gold particles. Then by using the, particle gun delivery method, these gold coated DNA particles are shot onto the plant cell. So because that complete procedure is under very high pressure, so because of that, these DNA coated with gold particles that will enter inside the plant cell chromosome and it will get injected. So once it is get injected, the transgenic crop would be formed. After that, these transformed cells are selected with the help of some selectable markers, and then transgenic plant can be consumed. This is the one way, that is gene gun. Another way is agrobacterium. That means we can do all these things in the bacteria also, because agrobacterium tumefaciens, it is a natural genetic engineer, in which what we do, we will be basically inserting this gene into the TI plasmid, and The bacteria would be mixed with the plant cells. The Ti plasmid will move into the plant cell and insert the DNA into the plant chromosome. So once this insertion is complete, after that the screening of the cell with transgene would be happening. That which of the plant cell has received the gene of interest. So as you can see, there are many crosses to it, but there is only one cell that has received it. This cell would be selected and transgenic plant would be regenerated from this single transformed cell, and then from there it can be used as edible vaccine. And what happens is, whenever you are taking this, whenever you are eating these edible vaccines, the first process is mastication and degradation in intestine. This complete thing happens in the case of pear patches that is present in the ileum of small intestine. At that time, in response to any kind of infection, the M cells that are present in the pear patches that recognize that antigen, it will differentiate, it will activate B cells to differentiate into plasma cells, and plasma cell will secrete IgA antibody, because the mucosal secretion only secretory IgA is produced. So as you can see, Pierce patches, rich source of IgA producing plasma cell would be produced. These edible vaccine will break down at Pierre patches, lymphoid nodules containing follicles for the development of germinal center. These antigen will penetrate the follicle and it will accumulate antigen in the lymphoid structure. The antigen will encounter with the M cells, which was basically expressing MSG2 molecules. The pocket formation would be happening in which B cells and T cells are already present. These M cell will activate B cells. It will differentiate into plasma cells at the malt region. Plasma cells will be producing IgA antibody, and this IgA antibody that is secreted into the lumen, it will interact with the antigen to neutralize it. So this is the complete mechanism of action of this edible vaccine. There are few examples to it. In case of banana, it is quite easily, transformation is quite easy. There is no cooking required. You can store it for longer time without any refrigeration. However, there are few limitations. It can spoil rapidly after ripening. It contains low protein content. However, it is quite a time-consuming task if you want to, have some edible vaccine prepared in banana. In case of tomato, it there are certain advantages. For example, it will grow quickly. High vitamin A content will also, provide an boost to the immune system. It is quite heat stable. It is tasting good. There is no special requirement for storage and transportation. However, there is only one drawback, that it may spoil easily. Next is rice. There is high expression of proteinaceous content. There is very low risk of contamination. Vaccine will not dissolve on exposure to some stomach acids also. However, there is only two disadvantages, that the growth rate is slow and you need special glass house facilities or special requirements to grow this crop. In case of maize, it is quite cheap. You need not to have some transportation or some refrigeration cost associated with it. However, there is need of some cooking before use, and the time taking concept is there. However, there are few more examples of it, of edible vaccine. For example, in case of hepatitis B virus, it has been used in tobacco, potato, and lettuce. And the antigen, the main disease that is being cured is hepatitis B. Norwalk virus, rabies virus, it was tried in tobacco. In case of coronavirus, transmissible gastrointestinous and coronavirus, it has been tried in tobacco and meats. The HIV virus, it has been tried in tomato. Vibrio cholerae, it has been tried in potato. So there are few patent holders of these edible vaccines also. There are many companies working in this area. The patent holder companies, Protgene, the recombinant antigen production and transfer to the plant cell using plasmid vector system was carried out, so as to treat the disease of hepatitis and enteritis virus. In case of ribozyme farm, nucleic acid vaccine was basically used to treat and prevent viral infection in plants, animals, and bacteria. Rubicon lab, the retrovirus was expressed in animal and plant cells, that was useful as virus of cancer vaccine. University of Yale, vaccine against invertebrate was prepared. University of Texas, hepatitis B virus core antigen recombinant vaccine was prepared. Institute Pasteur, that attenuated E. coli vaccine for use in gene therapy was being prepared. Vaccine was also prepared in transgenic plants by Biosem and Ron Merixes. However, there are many therapeutic molecules so as to that can be used to treat certain kind of condition. For example, insulin is a systemic factor. Glucose metabolism is the main target site to treat the condition diabetes. Erythropoietin, colony stimulating factors. There are many type of monoclonal antibodies also so as to treat these conditions. So these are the few MCQs pertaining to this current lecture. In case you people have any kind of query, you people can just by reading this you can get it solved. These are few MCQs that are regarding this lecture, that what was first approved vaccine, which kind of vaccine is known as live or killed vaccines, and who was the father of vaccination, who invented it, what is the difference between recombinant vaccines, subunit vaccines, toxoid vaccine, why there is a need, what kind of vaccine is being designed for COVID-19 also, that is the major hot topic right now. So that was all for today. Thank you very much for listening.